Okay, so today I'll be talking to you about these like spatial and temporal perspectives on dormancy with a slight focus on microbes. So far, we've heard a lot about how populations can benefit from dormancy and fluctuating environments as a method of bet hedging and kind of increasing the long-term persistence of those populations. But today I really hope to demonstrate to you, I guess, two sort of important facts that I consider in my work. And the first one of those is that these populations exist in a community context, right? So there are other species that are around in these environments that are potentially interacting with the focal population that could also have implications long-term for the persistence of those particular populations. And the second thing that I want to communicate is that these dormancy transitions and seed banking dynamics also take place in a spatial context. And so in addition to dispersing through time with through the entrance and survival and reemergence or reactivation from the seed bank, they're also potentially moving across space, and different organisms do so differently. And some of my key interests are trying to figure out how we can understand combinations of how species move through space and through time to better understand how they can persist in variable landscapes. So I think there might be a delay on this. So bear with me. Okay. So yes, so we often consider these processes like dormancy to be adaptations to variable environments, right? So they often average population fitness across space in the case of dispersal, or through time in the case of dormancy. And the basic premise here is we can increase the gains in the good times or the good places across the landscape and minimize the effects of the bad times, such that on average over time, populations have positive fitness. So an early model kind of getting at this was developed by Simon Levin and Dan Cohen and those folks in the 80s that kind of try to put dispersal and dormancy together in the same context. And so I'll briefly kind of walk through how they extended the classic Cohen model to incorporate dispersal, and then we'll see how we can move forward to incorporating more species to this model. So they were focused on population size of a species in a particular patch. And what they basically did was they broke it down into the population size now, the SIT, the fraction or times the fraction of germinants in the population multiplied by some yield function. And this function basically draws from a random environmental variable to incorporate noise in the environment over time. And this is multiplied by the non-dispersing fraction. So if there's a fraction D of the population that's dispersing, then this germination and yield function operates just on those that stay locally in the patch. And then to that, we add in the non-germinating fraction of seeds. So if G is the germinators, we have the seed bank that survives at a particular rate. And then we also incorporate influxes from the rest of the metapopulation. So we have some dispersal success parameter here that incorporates how likely migrants from other patches are likely to end up in a focal patch. And this is weighted, of course, by the germinating dispersers in other patches. And this is averaged by the yield of those other populations in the metapopulation. And so this is sort of an early approach to try to get dispersal and dormancy in the same types of models. And they've refined these models over the years, and people have continued to work on these questions, but typically it's remained in a single species context in this case. But what they really found was that the optimal strategies for dispersal depended a lot on how successful migration was in the environment, so that immigration success parameter, as well as how likely like dormancy was to actually occur. So there's this interaction that they found between dispersal and dormancy that could potentially alter the successful strategies that emerge in this metapopulation. So key point here, dispersal and dormancy are not independent, and they can have these interactive effects on populations. So today what I'll do is I'll tell you about some models that I've been working on lately to start to investigate the spatial effects of dormancy in a community context, so multiple species. And then I'll transition towards some more empirical work that is sort of documenting these patterns of dispersal and dormancy in a natural system. And then I'll talk about some future ideas and speculate with some potential topics we might want to explore in the future. So I mentioned that dormancy occurs in a multi-species context. And from this, I'm going to draw on metacommunity theory. So metacommunity theory is probably, I would say, one of the leading frameworks in community ecology to incorporate spatial dynamics into our understanding of biodiversity maintenance and the dynamics of multi-species communities. And basically what it does is it breaks down community dynamics into local scale processes and connections between those local patches across the landscape via dispersal. So one of the factors is the fact that different species respond differently to the environment. Right? Let's say we have some abiotic gradient here on the x-axis. This could be a temperature gradient, a precipitation gradient, a pH gradient, some type of environmental axis. And different species are likely to have maximum growth rates that are distributed along this axis in different ways, right? So some form of niche partitioning in the environment. But on top of this, species also interact with each other, right? So species place limits on their own population growth rates through competition with their other members of the same species, but they also interact with other species in the environment by taking up resources that are shared between them, by directly competing them, by eating them sometimes, or by parasitizing them, or they can even provide positive interactions through mutualism and facilitated interactions. So we also have to incorporate these density dependent effects that change with the size of the populations that are actually being modeled. And finally, we can embed these density independent and density dependent processes in a spatial context by linking patches together with dispersal. And so our goal with our project was to put a seed bank on this. Like what happens to metacommunity dynamics when you start incorporating local scale storage in different habitat patches across the landscape. And so we built this spatial seed bank model mostly from an annual plant perspective. So these models have been pretty well developed, and they extend from Cohen's work into a more multi-species application of Cohen's work. And so we wanted to embed these multi-species seed bank models into a metacommunity framework. So what we've modeled here is we've taken the classic model of seed bank survival, germination rates, production rates, but we've now coupled them with immigration and immigration across the landscape. And this gets then embedded in a spatial network. And so we've also taken into account a spatial explicit approach to our modeling. So the patches actually have a spatial identity. So nearby patches are actually more connected than far away patches are. And I'll get into how we compute that later. But the idea here is embedding these seed banks into each of the local patches. So briefly, what our model, how our model works is we just created a discrete time simulation to capture the annual dynamics of these plant communities and variable environments. And the change in a population size in a given patch X of species I is simply the survival rate of that species in the seed bank in that patch, the production of new seeds in that patch, the immigration of individuals from that patch to the other patches in the landscape, and then the immigration from all those other patches into the focal patch. Right. So we modeled seed production as a product of germination of that species in a local community. times its above ground growth. And our germination was modeled stochastically such that some per capita germination probability, we draw basically a binomial, or from a binomial distribution to get the number of germinating individuals at a given time step. And so this is capturing this reactivation parameter here. And then we modeled the above ground growth dynamics through what is classically called a Beverton-Holt model in population ecology. And basically this breaks it down into the density independent effects that we talked about before. So how much the environment limits your growth, rescaled by the density dependent effects. So stronger competition in this case would more strongly reduce growth rates. Yes.
So what you're recording here in the abundance is really the number of seeds, right? There's no explicit modeling of the active population, right? It's implicit, right?
It's implicit, but we kept it separate in our computation, but you add them together to get these dynamics. But these different terms are taking account of different above ground versus below ground populations. So in our models, we actually do have them separated. Yes, yeah. So this here is the total number of above ground and below ground.
Oh, the total.
Okay,
So the seed survival is the soil, and seed production is really not only seeds but also regarding seeds.
So you're right in that it's only capturing the seeds here, but in the simulations, we keep track of how many germinate per time step and how many seeds are produced per time step. So we actually have that distinction in the computational side of things, but for this notation here, we don't have it separate. Does that clarify? Okay, yeah, thanks. There's not so much a binomial or a biological motivation as in it's somewhat of a simplistic way of thinking about it. So we're not incorporating any kind of dependence for germination on any other individuals in the population. It's sort of just, if we grab it this many times, how many are we getting with some kind of noise distributed around it? That's a good question. Yeah.
So every generation is individual. For instance, every individual will decide who's in aquatic, it will be a seed or
basically, yeah. So for every individual decides basically, or every individual in the seed bank. So yeah, every individual decides if it's going to germinate basically from the seed bank because it's an annual plant model. And so the assumption here is that at the end of the year, there's nothing above ground, it's all in the seed bank. And you're gonna grab from the seed bank and say who's germinating. And then you're gonna figure out of those germinators, how much are they growing? So these effects here are mediated just by the germination fraction. So there's some different like sequential stepwise effects here that I think are hard to convey in this way, but these things do happen in a sequence. But yes, so every year the population above ground goes away and we reestablish it from the seed bank. Does that make sense? With some stochasticity that creates some variation in who gets to activate and establish. But yeah, so the density dependent effects here are modeled as these alpha terms, which are just basically competitive interactions between same species and between other species. And these are weighted in the denominator here by the germinated fraction of that species. And it's weighting its effects on a focal species. Yes.
So all the types of you listed us.
Mhm.
variety of interactions. And I'm surprised that you could synthesize that with a linear combination only. I mean,
True.
it's very strong. But if you say
Yeah.
it's efficient, I believe you, but I don't know what.
No, that's a good point. And in a lot of these community ecology models, there's been decades of debate over whether or not like these linear terms are sufficient to actually capture true dynamics, or whether we need to incorporate some of the more higher order terms. And an ongoing debate, I would say. But I think for a first pass, especially with maybe more simple interactions, this can be sufficient to get some first ideas out there. And in this case, I'll only be measuring competition. So it might be a simpler way of capturing that. But if you were to get into, say, some like predator-prey models, you have these functional responses that have saturating effects and nonlinear effects as well that you might want to incorporate if you were to get those more well-described interactions. But here we're just assuming linear effects of one population on the growth rate of the focal population. But that's a good question. Yeah. And so for the density dependent or independent terms, so this is where we're modeling the effects of the environment on the growth rates. All species have some maximum growth rate in population, and we're just decreasing that with a negative exponential away from their optimum environmental condition, weighted by some niche breadth. So they can have a wide niche breadth or a narrow niche breadth. We weren't interested in that at the moment, so we kept those all the same, just to get some idea of how distribution along that gradient affects outcomes. And then what we did was we incorporated demographic stochasticity in the model. So what we calculated before, that's sort of an idea of what could happen potentially, but in populations, there's random fluctuations in births and deaths that could potentially increase or influence the ultimate number of births that occur in a population. So we basically just drew the number we calculated before from a Poisson distribution, or zero if the population went extinct. So if those previous computations shifted the growth rate to be, or the population size in the next generation to be negative, we took it to be zero, or we drew it from a distribution to get some fluctuations. And then for seed survival, we kind of did the same thing as we did for germination, except now we're focusing only on the non-germinated fraction of the population. So we have our total population size minus the germinated fraction, and we drew that from binomial distribution with some per capita seed survival rate. And this affects the carryover between generations. And then we modeled immigration the same way, basically. These are all individual decisions that are occurring in the model with some probability. And we're modeling germination or immigration on the germinated fraction of the population. So we're assuming that if you're in the seed bank, you're buried enough to not get dispersed around. But if you're still on a plant, you can potentially be blown around and disperse at some probability, where D is the immigration rate. And then immigration then works on that pool of migrants. So all the patches contribute emigrants to a larger migration pool. And then we basically redistribute those across the landscape in a spatially explicit way. So the numerator here basically is all the migrants from all the patches that are weighted by the distance from that patch to the focal patch. So this distance term is here in the negative exponential. And this is just a ratio to all the migrants in the whole pool that dispersed at a given time step. So this gets just redistributed proportionally. And what we did was we basically generated some stochastically varying environments that incorporated spatial heterogeneity among patches. And these were modeled in such a way that they were spatially autocorrelated. So nearby patches are more similar than others, but there is some variations. It's not perfectly constrained. So yeah, so we that was something we varied as one of our like simulation experiments we run. But they were the same across species for now. So we're still working on these projects, getting into the ideas of trade-offs among species and seeing how species with different trait values and different parameter values might influence their coexistence or not. But for here, we're going to basically simulate it across that parameter space and see how changes in those parameters influence patterns of biodiversity and species richness across different spatial scales. But they're all the same for now. Yeah. And these environments then fluctuated through time in ways that were, again, autocorrelated to kind of incorporate seasonality. But because of the spatial heterogeneity, there's a sort of acyclic or asynchronous fluctuations across the landscape that are occurring in a way that is correlated across space. So basically conditions that should favor dormancy and dispersal. We put 40 species on here to start with, and we simulated this for 2,000 time steps. So some long-term maintenance of diversity is what our interest is in this case. And then we model different types of species interactions. So in the one case, we made all those competition coefficients more or less equal. So they're basically, there's no difference between competition within species and competition between species, which shouldn't be a stabilizing effect, right? So if there's no differences in how species are using the landscape, there's no way for them to kind of create conditions that should favor coexistence among them. But we also incorporated the stabilizing conditions, which is basically when competition between members of your own species is stronger than competition with members of other species, which creates this self-limitation and negative feedbacks that stabilize population dynamics and can eventually facilitate coexistence over the long term. Yeah, and then we just vary dispersal rates, germination, and survival rates. So one of the things I'll talk about is how dormancy affects diversity across scales, and I thought I would demonstrate what I mean by that. So we have local communities consisting of many local populations that their diversity within a particular community can often be considered alpha diversity. That's kind of jargony from our field, but basically whenever I refer to like local diversity, it's the diversity you're finding within a particular community. And then we can look at differences in diversity among those communities. So how much is this spatial turnover between them? And this is often called beta diversity. And this is just getting into how different is one community from another community. And you can see that if you have really high local diversity and really high turnover amongst communities, that product can actually scale up to increase diversity at larger spatial scales. So if you look at the collection of those, we're looking at the regional diversity or meta-community diversity or gamma diversity. And so these local diversity times turnover is an important factor in maintaining diversity at large spatial scales. And so there's sort of this iconic pattern in meta-community ecology that I've sketched out here, where as you increase dispersal rate, you get this sort of pattern where the diversity at local scales shows this hump-shaped relationship. So at first, you're able to push more species into local communities, their diversity goes up. But at some point, they start competing too strongly and you kind of converge on this regional low diversity dominating species, and so the diversity plummets. The differences among sites always decreases with dispersal because you're just homogenizing it, right? So if you have separate populations and separate communities, mix them all together, they all become the same. And then the regional diversity also shows that decreasing relationship as you increase dispersal rate. So I'm showing you this because this is a pattern we're going to look at with seed banks as well. And we're going to see how seed banks can potentially modify this pattern. But first, we just wanted to show, can we actually reproduce this pattern in our model just to make sure that they're capturing what we know is pretty common about communities. And it turns out, yes, we can also recreate this in our models. So we find without a seed bank, basically the same kind of patterns, just different scales. So now let's focus on that top panel and see what happens at local scales when we add in seed banks. And so what we find is that if we're looking at, again, dispersal rate on this axis, and on the y-axis here is how many species on average are in the local community. So each of these little dots here is one simulation. So there's lots of replication for each parameter combination. And I'll show you different colors of lines here that match up to different germination rates. And so when we look at the effects of germination rate here, so if we have our no seed bank condition in this orange line, we can see that as we decrease germination rates or increase the probability we have these large seed banks, we can actually maintain something crazy like 3 or 4 times as much diversity on average in local communities in a way that depends on dispersal rates. So there's multiple ways to maintain diversity in these systems. And sometimes it's could be no seed bank and intermediate dispersal rates, or the same diversity can be maintained by having some seed bank with a super high dispersal rate, or much more diversity can be maintained by having a very big seed bank and some kind of higher level of dispersal. So there's interactions emerging between these two processes that can influence patterns of diversity in ways that differ quite dramatically from the case with no seed bank. Jay, yeah.
There's a general question about the models and the number of species that you're seeing actually. So
Yeah.
on the left-hand side, the truth that there's somewhere on 1, 2 species total in a patch. That's not correct. Why can't there be more in a patch?
Yeah, so this is that is accurate. So for much of this parameter space, we have 1 species because it's just whoever is there, and there's not enough dispersal to actually get them anywhere else. And each species is best fit to 1 patch, basically. In some cases, we have like 4 or 5. But this is the condition that I should have mentioned where there are no stabilizing interactions. So we would actually expect there to be quite low diversity. Sorry, I should have mentioned that. So yes. Yeah, and most of those species that you create just can't persist in this landscape. A lot of them get lost. And what happens is this is just the ones who can coexist by partitioning the environmental heterogeneity in the landscape, but they don't actually interact in ways that stabilize their coexistence. So if we look at spatial differences, so this was that among site turnover. Again, we see our typical decline in the absence of a seed bank, but when we add seed banks, we don't really see that strong an effect. So there are like slight increases, but it's really not that big of an effect in this case. The biggest effect seems to come when dispersal might be somewhat limiting in the landscape, and then having a seed bank on top of that actually does an inhibition of turnover. So not a ton of effects in the spatial case at the moment. And regionally, this translates to at the regional scale, pretty big increases again in the regional diversity at the metacommunity scale when you have low germination rates and these intermediate to high dispersal rates. So again, the effects of the seed bank depend on the dispersal rates in these models. Yeah.
So are these results robust when you change the population size?
We didn't really vary the population size. We calculated that in response to a reviewer, but they were, I think, around the size of like 40 or 50 individuals usually per patch or per local population. So pretty out of the range for stochastic extinctions occurring, but they're not huge populations, but they're somewhat reasonable. And if you were to change that, I mean, I think it might just shift things down in a way if you were to get smaller populations. because you would increase more of those stochastic effects that are occurring that lead to decrease.
If you have a big seed bank, how you increase the technical risk, rather than some compounding.
Yeah, we found that because of the way that we modeled the seed banks in more of like a bet hedging way, where there's just sort of this rate that's changing back and forth. It's not dependent on the environment. Those population sizes actually look pretty comparable. So the above ground population and the below ground were actually kind of on the same scale, and we didn't get like rapid increases in the seed bank that are humongous. They were actually cleared out at a pretty constant rate. But yeah, it's a good question though. I think that would be neat to explore the population size effect of that and see if these things change at all. Yeah. Any other questions? Okay. So then we look at the effect of species interactions, right? So I mentioned to you that we have certain conditions we know from ecology that can potentially increase coexistence and set up conditions where species are able to persist together. And can those interact with the effects of dispersal and dormancy in ways that also affect diversity? We found kind of different effects, right? So when we change the structure of our competitive interactions, we find this type of relationship now in the absence of the seed bank. And so basically that's saying is when you set up conditions that favor coexistence, increasing dispersal actually can just allow more species to coexist locally because they have those stabilizing interactions. And so you're removing any barriers to colonization when you increase dispersal rates, putting species together in local communities, and they have actually end up coexisting. Interestingly, we find that seed banking in sort of this case where survival is a bit riskier can actually have inhibitory effects on diversity at the community level, which caught us by surprise a little bit. And we think what's happening here is that in this case where species have the potential to coexist because we've set them up to do so, remaining in the seed bank that's risky is actually causing losses of diversity in local communities because it's inhibiting them from colonizing the above ground and actually coexisting and making new seeds, right? So if they stay in the seed bank and it's risky, it's more likely they'll actually get lost through the seed bank process than grow above ground and make new seeds to replace it. So sort of some unintuitive results at the community level for that case. And if we really increase survival a whole lot and make it not as risky, it all kind of evens out in a way. And now when we look at turnover, so this is again the differences among communities, we find that the biggest effects here of seed banks occur when survival is, again, kind of risky in the seed bank, but dispersal is potentially limiting in the landscape. So what we think is happening here is that when dispersal is so low that species can't track their favorable environments across the landscape, that seed banks are really important for basically allowing populations to persist in their favorite habitat and continue to maintain this spatial heterogeneity and community composition across the whole landscape. And if we look at how this translates up to the regional scale, so the large scale, what we find is that, again, in this risky case, there are conditions where having really high dispersal, but really low germination rates in a risky seed bank can actually inhibit the diversity we see at the regional scale, which is kind of intuitive for, I think, many ecologists to think about, where we often think seed banks are always good in that case, but it can actually impose a demographic cost on lots of populations, especially if they should be good at coexisting. But again, if we increase the survival rate of the seed bank and make it not quite that risky, we find that there are pretty pretty much consistent benefits of seed banking across the entire dispersal gradient. Yeah, has pretty much always maintained diversity in that case. Okay, so just to summarize the main take homes here that I wanted to say. Yeah, so dormancy can influence population persistence in ways that actually do appear to depend on rates of dispersal in the landscape. And the types and strengths of species interactions can also influence the relationships we see between dormancy and dispersal and diversity. Now I'll move on to some brief stuff that we've done in the field. So a lot of what we talked about so far has been sort of hypothetical, but as microbial ecologists, we've been really interested in trying to detect these signatures of dormancy and dispersal in different environments for some time now. And I'll show you a few things we've been learning from my work that I've been involved in. So just to demonstrate, microbes play pretty pivotal roles in aquatic ecosystems. They're important decomposers, and they're important for making nutrients available to other species in the environment. So again, they're kind of a crucial part of microbial or freshwater ecosystems. And one question is, do dormant seed banks appear to help maintain microbial diversity in freshwater systems? So this is a picture of a lake near Indiana University that we've been looking at for years. By we, I mean Jay, trying to understand the microbial dynamics in this particular ecosystem. And one of the things we did was basically went out and took water samples. And we split those into what is the microbial DNA we find in this particular sample, which tells us something about which microbes are present in that particular ecosystem at that point in time. And then we looked at the RNA transcripts. And so this is a closer indicator of who's actually reproducing in that system, which tells us a bit more about who might be potentially metabolically active in that particular environment. And then we basically just compared the dynamics of these two different pools to get some idea of to what extent the total community or the DNA community is actually elevating diversity relative to the active community. And so a simple way to look at this is just what is the total richness or diversity that we find in a sample at a given point in time where we're measuring the total diversity to the active diversity. So higher up in the graph means that there's more diversity stored in the seed bank relative to the active community. And then below is the opposite. And so over the course of 2 years, we found something that I think is quite interesting. There's a seasonality to the seed bank. The richness that's being stored in the seed bank appears to fluctuate quite a lot through time, and does so in a way that tracks environmental conditions, right? So in winter months, we have peaks in the diversity that's in the seed bank relative to the active community. So maybe there's some role here at the seed bank for maintaining diversity during these unfavorable periods in the environment. So I think what this helps us see is that this is a dynamic, like the seed bank is a dynamic system, right? It's not just a static pool of diversity to draw from. It's actually changing based on inputs and outputs. And these rates can vary through time in ways that influence the composition and potentially the size of the seed bank over time. So I think incorporating that dynamism into the seed bank conception is actually, I think, really important. Yeah.
So the value change, but the total richness was the same.
Yes, it does, but the change in the total community is dampened relative to the change in the active community. And so I think what's happening is that the changes in the active community are driving some of this pattern, but it's the ratio. It's telling us something about like the importance of that seed bank for preserving that diversity over time. Does that make sense?
more or less quantum the real changes.
So the diversity of the total pool changes, but it changes, I think, if I recall, it changes less than the active pool. The active pool changes as well, but it changes more. And I think that's what's driving this pattern. In terms of the actual like rates of inputs and outputs, we don't know. That'd be really awesome to know if we could actually track those transitions. But that's something that I think we don't have the tools to do yet. But what we really need if we could actually put some ideas of those transitions into our conceptualization of this process. But yeah, did that answer your question at all? Or, okay. Another thing to keep in mind is that in these aquatic systems, they're actually open systems, right? So you can also think about this from a metacommunity perspective, right? There's immigration coming into these local aquatic sites, and that can also potentially influence the diversity we find in a particular site. So so there's a picture to show you. They're pretty intermingled sometimes. And previous researchers have found that they detect quite a lot of terrestrial derived bacteria. So they think that soils are contributing a lot of diversity into aquatic ecosystems like streams, rivers, and lakes. And so we wanted to know, well, are those microbes coming in actually doing something? Are they active or are they dormant? So we go back to this sort of canonical figure that everyone's included so far. Look at the rates of dormancy on freshwaters and soils, right? So soils are super high. So a lot of those terrestrial derived bacteria, they could be dormant. We're also seeing pretty high freshwater dormancy too. So to what extent might those migrants coming in be metabolically active and contributing to ecosystem functions versus just passing through the system as dormant individuals? So we went back to the same lake we were at before. This is just an aerial view, and we sampled along a transect. And we captured the soil samples here and the influx and outflux of this particular water body along that gradient. Did our same splitting process, so we have some idea of who's there versus who might be metabolically active. And what I'm showing you here is the richness we estimated through some process of the bacteria in the system along this reservoir. So this is where the soils are coming in, and this is where the water is flowing out over the dam. And what appears to be happening is there appears to be this really big increase, like threefold increase in diversity near that point where the soils might be coming into the lake. So we basically captured the same pattern that everyone else found. So we have this high diversity, and in other graphs that I'm not showing you, this looks a lot like soil bacteria, and it's coming into the system. But if we look at just the metabolically active subset, it's actually really low. So it appears that there might be some split between who's coming in and remaining dormant versus who's actually growing and actively reproducing in the lake. So it seems like there's another interaction here between dispersal into the lake and dormancy that is having effects on spatial patterns of diversity as well. And so it seems like dormancy is almost facilitating this migration and persistence to some extent into the lakes. But this remains kind of localized to the perimeter. Like as we go further into the lake, those effects disappear. So there's some spatial aspect of that.
Have you ever thought about, I should know the answer, putting these two projects back to back, and you just showed us that Z-bank ratio.
Mm-hmm.
Have you ever thought about the plotting, taking these data and plotting in that same measure?
I haven't, because that one came after this one. But
I suspect that you would see the Z-bank contribution to both richness, decrease.
yeah, I think it would track this pretty well. Yeah. And that would take into account the identity of the species, but it's a good idea. We could check that. So just to kind of sum up some of the insights from empirical work, this includes some things I didn't show you. But basically, it appears that few taxa, like few species that are coming into the lake, actually established in the lake from the soils. And it seems like most bacteria coming in that have a potential soil origin are actually remaining inactive and just passing through the system. So there's more graphs I didn't show you for that, but the diversity effects then appear to be quite localized towards that boundary of the lake. And over time, it appears that dormancy can actually help maintain some lake microbial diversity, in particular by preserving diversity over harsh winter environments. And we also have some evidence suggesting that this dormancy could be important for basically allowing populations to persist over the winter and facilitate recolonization in the spring bloom that comes out after ice melt. So to me, this suggests a couple questions, right, that might be worth exploring. So I think in systems with many species and that have dispersal and dormancy, and what are the relative contributions of dispersal and dormancy to different mechanisms of coexistence in these systems? Is dormancy sufficiently strong? Do you need interactions between dispersal and dormancy? And if the seed banks appear to be seasonal, is dispersal seasonal too, right? Like you can imagine different patterns of rain that could affect dispersal rates as well. And so modeling dispersal and dormancy as a sort of dynamic rates might be interesting. And then how about the interaction between these rates, right? Like, are they synchronously going up and down together? Are they compensating for each other in different periods of the year? And then we talk a lot in ecology about dispersal kernels, right? So what is the distribution of potential dispersal rates in a particular population across a landscape? Are there dormancy kernels, right? What's the distribution of time that individuals can remain in the seed bank? And how does that distribution, particularly the long tail of that distribution, how does that influence the dynamics of the system when you can have these really persistent individuals that can emerge way off in the future? And does that shape influence the system at all? And so another theme of this workshop was host-parasite dynamics. I think there's a lot of relevance here to that as well. So, you know, like we have dormant pathogens, we've heard a lot about that, but about dormant hosts, you know, is there an interplay between dormant hosts and dormant pathogens? And can we examine that aspect? And we've often assumed, including in my models, that dormancy is constant rate. But some of the work that we've seen here suggests that there may be also responsive changes to these rates. And what happens if you kind of couple these responsive switching with that hedging strategies and kind of get these really more realistic, intricate mixed strategies emerging? How does that affect community ecology beyond population ecology and fitness? And based on some of these results, like, can dormancy actually be a way to store genetic diversity in unfavorable habitats, right? Like, can you use the space, like on the landscape, as another mechanism of storage for seed banks mediated by dormancy through that interaction? And just some broader themes more generally, I think it'd be interesting to think, for example, in our group, like, what about dormant seed banks beyond just simple competitive explanations, right? Like, there could be predator-prey explanations for why things go dormant. There could be more complex food webs we can create that incorporate dormancy. And there could be lots of implications for dormancy that have broader effects. It could affect transmission. And this also plays into these broader models of dormancy or of adaptation across space, right? Like, so we're talking about genetics of host-parasite interactions. There are lots of conceptual theories that we can draw from, like the geographic mosaic theory of coevolution, and even some more quantitative stuff like metapopulation theory. How does dormancy and host-parasite interactions fit into those models of local adaptation? And can we better understand the role of dormancy in mediating adaptation, depth dynamics, drift across these bigger scales of space? And with that, I have a lot of people to thank, including all the co-authors on this work. Well, okay, one extra one pop in there. and all the funders for helping support these projects. And with that, I'll take any questions if you have them, if I have time. Thanks.